Okay, last time we talked about how bacterial chemotaxis works, the biased random walk that bacteria do towards food or away from toxins. Today, what we're going to do is talk about the mechanics of this, the pathway involved, which is what we're going to focus on is the chemotaxis operon, which has several proteins involved. These are all kind of given letters. And they all start with key for chemotaxis. There's key Y, key P, key B, key R, and then also key Z and key A. We'll talk about all of these and their functions in turn. But the starting point for chemotaxis is actually the receptor protein, the receptor molecule, which is a transmembrane protein, which has a domain that sticks out of the membrane, which we call the ligand binding domain, and this binds different ligands, which can be amino acids like aspartate and serine, which the bacteria likes, or it could be sugars like maltose. These generally bind to the receptor with kind of an adapter protein, a dipeptide, which binds the sugar, which then in turn binds the receptor. So there are, I think, 5 known receptors. There's TSR and TAR, which are the serine and aspartate receptors. There's TGR, which is for sugars, like I said. There's TAP, and newly discovered AER, which is for oxygen. The bacterium has a great number of these receptors distributed across its membrane. As an example, if you look at 3 strains of E. coli, we can see that the single cell can have up to 40,000 copies of this receptor embedded in its membrane. Interesting thing is, if you notice that if the environment is rich in sugar, it tends to have less expression of these proteins, these receptors, which is a form of adaptation or kind of optimal expression of a protein, which we'll get to in later weeks or later when we talk about optimality in biological systems. So there's nominally about 10 to 20,000 of these distributed across the membrane. Inside the membrane is where the interesting stuff happens. At the bottom of the receptor, there are binding sites for the key A and key W proteins. Key W doesn't do much. It's a connector protein that connects the receptor to key A. The key A is the primary kinase that phosphorylates several different proteins, most important of which are key Y and key B. Key Y is the primary activity protein in our case. We'll later see that when you get more phosphorylated key Y, the more likely the bacteria is to tumble. So the tumbling frequency is directly connected to how much phosphorylated key Y, namely key YP, there is. And key B is demethylase, which will demethylate the receptor when it is phosphorylated. So its active form is key B phosphorylated, which is actually getting phosphorylated by key A itself. So we'll see later on that key A is actually activating its own inhibitor in a sense. Another key part are the methylation sites on the receptor. These sites are places where key R can add methyl groups. It changes from receptor to receptor, but there are typically 4 to 5 methylation sites on each receptor molecule. And you can think of these methyl groups as enabler buttons. So as they get added, so as the protein gets more methylated, it becomes easier for the ligand to bind, but also it becomes more active with respect to its phosphorylation effects through key A. So the more methylated a receptor is, the more likely the ligand would bind to it. Also, it's more likely that key A will phosphorylate either a molecule of key Y or key B. So if we take the whole pathway as a whole, as you can see in the figure, receptors shift between different levels of methylation, and they could be demethylated completely or methylated completely or somewhere in between. To keep things simple, we'll say the methylation amount is going to be either 1 or 0. So we'll talk about active receptors versus inactive receptors. In truth, that's not the case. A receptor could be partially methylated, in which case it's actually partially active. But just to keep things simple, we'll say it's either methylated or not. The other aspect of this is the attractant, or the input to the receptor, the ligand. Now, the thing is, when the ligand actually binds to the receptor, it inactivates the receptor, whether it's methylated or not. Higher methylation also means that the attractant has an easier time binding, but what's important is if a receptor is completely demethylated, doesn't have the methyl buttons on it, then it's completely inactive. If it's methylated, it becomes active, meaning key A attached to it can phosphorylate both Y and B. But if it's methylated but also a ligand is attached to it, it becomes inactive again, which means the phosphorylation stops. Now let's see what this means. Key A phosphorylates key B, which means as there's more active receptors around, so more of the methylated unattached receptors, the faster key A is phosphorylating key B, which means there's more phosphorylated key B around, which in fact demethylates the receptors, making them inactive. So there's a negative feedback loop there. The more active A means more active B, which deactivates A. R, on the other hand, is relatively stable. It's not affected by the state of the receptors. It constantly and in a kind of a steady speed methylates demethylated receptors. So it's like a little engine that never stops. It's slow, but it's steady. It will methylate receptors at a certain rate. B, on the other hand, depends on it being phosphorylated or not. So if there aren't any methylated receptors left, soon most of B will be dephosphorylated or inactive, which means it won't be demethylating or erasing those little M's off of the receptors. So if that was the case, in time you'd have all the receptors fully methylated. On the other hand, if a lot of the receptors are methylated, then B would be more active, and then it would demethylate at a rate faster than R, which means you would shift towards more demethylated receptors. In normal case, at steady state, the rate at which R methylates receptors gets equal to the rate at which B demethylates receptors. So there's a steady state fraction of receptors, or steady state amount of methylated active receptors on the membrane. The other side of this coin is A also activates Y, key Y. So what happens there is when A is active, meaning the receptor it's attached to is methylated, but not bound by a ligand, what's going to happen is it's going to phosphorylate Y, which will cause the little motors on the flagella to cause the bacteria to tumble. So the more phosphorylated Y there is, the more tumbling there's going to be. So as the amount of phosphorylated Y increases, you're going to have more tumbling activity, and that's what we're interested in. Key Z is the equivalent of key R for Y. It will constantly and steadily dephosphorylate Y and is not affected by the receptor state. So let's look at the dynamics of this. In this kind of pathway, we have first and foremost the kinase A, which will activate Y, right? And also we have B, which demethylates A, and we have our signal, which is the amount of attractants A. And what else is there? There's Z, and also R. Did I miss anything? No. Now there are certain relationships between these, and I'll draw them in different colors and I'll explain what they mean. So first of all, let's go red, and here we have A, which activates Y, obviously, and Z, which inhibits Y. We also have the signal, the amount of attractant that binds to receptors, which inhibits A. So when the attractant gets less, A is slows down as an activator. And then also A, when it's active, it will activate B, which will in turn demethylate A. Now, the second set of reactions is where R, which constantly methylates A, and also B, which demethylates A. Remember that the more demethylated the receptor is, the less active A is. So these are methylation reactions. In this case, demethylation. And this one is phosphorylation. This one is phosphorylation as well. This one is dephosphorylation. And finally, the signal is essentially binding, right? So the reason I draw these in different colors is the red ones are fast reactions and the blue ones are slow. Meaning, usually what happens is once you have new receptor binding to A, the level of active B, phosphorylated B, jumps up or goes down very quickly. Whereas once the activity of A increases, the amount of demethylation goes slower. So this is kind of what happens. Now when you look at, let's think of this as a chart. Let's say we have something like this. Over time, okay, and now A, if you remember, is essentially the amount of methylated but unbound receptors. So the activity of A depends on receptors that are methylated and the fraction of those, let's call that small a, that are unbound. meaning they don't have any ligands on them. So let's say the amount of signal affects how much this activity is. So let's say we have a certain amount of sugar, and at one point it jumps up and then goes back down. So that's S. When you look at small a, it's going to do the opposite, because since binding is fast, the moment the amount of sugar increases, some sugar molecules there'll be sugar or amino acid, doesn't matter. So let's say the attractant, when it increases, it will be more likely that it will bind to A, and that binding will be very fast. So let's say A, remember, is the fraction of unbound receptors. So when there's less sugar, let's divide this into 2, there'll be more activity. And then when sugar increases, the amount of unbound receptors will drop. And then when sugar leaves the area, the receptors will get unbound again. So the activity is kind of the inverse of sugar, right? Rather not the activity, but the fraction of active methylated receptors. So now let's look at methylation on the same graph. Okay, so that's a bit crooked. Initially, this is the receptor activity, the small a, and let's take a look at Y, for example, which is our tumbling activity, right? The more Y there is, the more YP rather there is, the more tumbling there is, right? So initially, when there's the receptors are highly active, you'd have more tumbling. So that's Y. And when the activity drops, the tumbling rate drops as well, very fast. But what's also going to happen is the amount of B phosphorylated is going to drop very fast as well, because B is obviously being phosphorylated by A. In time though, since R is working slowly to methylate A, even though the amount of unbound receptors doesn't change, more of them and more sites on more of them will get methylated. But remember that this is slow, so it will happen slowly, and Y will come back up in time, hopefully to the same steady state, which means your tumbling activity will first decrease as YP goes down. Bacteria will tumble less, but then in time, as more and more of the receptors get methylated, it will compensate for that, and the tumbling will come back to its original level, because YP will have come back to its original level, because even though A is lower now, XM will have become higher, the amount of methylated receptors, and you'll get essentially the same output. Now, this is the ideal case. This is what we're looking for, that it starts off from some steady state, adapts or reacts rather to the increase in sugar, but then in time kind of adapts. This is kind of analogous to, say, how your eye works, right? Remember that, for example, in your eye, you have your iris, which has a certain kind of thickness at any given point. And what happens is, as, for example, there's suddenly you walk into a bright room or a dark, let's say you're in a bright room, you suddenly walk into the dark, you can't see anything. In time, your iris expands, opens up, letting in more light, and now you can see what you couldn't see before. Methylation is like the iris here, in that initially it had adapted to some level of sugar, but when suddenly the sugar increased, the amount of unbound receptors dropped. Now it's basically methylation is increasing slowly, xm, which is the amount of methylated receptors, which is you can think of it as the sensitivity of the system. So in time, the iris opens up, the sensitivity increases, and you come back to where you were. And now when the opposite happens, so let's see, this is the point. Suddenly, now, what's going to happen is, as the sugar goes back down, the amount of unbound receptors will jump back up, meaning A will suddenly become more active, meaning it will increase tumbling because of it will phosphorylate more Y. So suddenly your activity jumps back up. And in time again, since the activity of A also increases the activity of B, B will slowly shift the steady state and demethylate more of the receptors. So sensitivity goes, well, not like that, but it goes back down again, hopefully to the same steady state, which means the iris has become smaller again. So this is typical in many systems where you have fast reaction to some parts and slow compensation. You can think of this in terms of your own home. For example, your heating system. The heat slowly drops because you open the window, or it's the evening now and the heat has dropped. At one point, your thermostat decides to turn on the heater, and that's fast. The turning on is an electrical thing, so it's fast to do that. And slowly now, the heater comes in and it heats your house, and at one point it shuts off because it's at the right set point, which is again a fast control. So it's a slow system with fast controls, and you have this negative feedback here, which allows you to, in essence, have homeostasis. The first model we're going to look at is the fine-tuned model, as it's called. It's a model that's based on this understanding of how methylation works to sensitize and desensitize receptors, therefore the activity of key A, which leads to more or less tumbling for the same amount of attractant, and we'll build the differential equation system with respect to this. Notice that the differential equation only involves the methylation reactions. We said before that the phosphorylations, remember the red arrows, are very fast with respect to these. So the methylations happen on the order of seconds or minutes, whereas the phosphorylations are maybe a tenth of a second or so. So from that time scale, the time scale of methylation, the other reactions are instantaneous. So we'll reflect that as the sudden change of velocity in the demethylation effect of B, namely VB. So let's take a look at this equation part by part. The first part obviously is the rate of change of methylated receptors. So X, let's say, is the concentration of receptors, and therefore XM is the receptors that are methylated. So dXm over dt is the rate of change in the fraction of methylated receptors. VR is the rate at which R works, meaning adding methyl groups. We said this was steady, and for the beginning we'll take this whole term VRR as one constant number. And notice that R is a relatively less abundant protein, so there's a constant number of those at any given point in the cell, and they work at a steady rate rather. B, again, there aren't too many B's, but the rate is dependent on how many of them are phosphorylated. So more B that's phosphorylated, VB is effectively faster. And finally, we're assuming that B acts on receptors in a Michaelis-Menten type equation, and that the substrate is the receptor and B is the enzyme. So it depends on the Michaelis-Menten equation with constant K. This could also just be a first order equation, so you could just write this as VBB times XM. And most of the time in the mid regions it acts like a linear equation, since the middle of the sigmoid will be more or less a linear function. And we'll see later on that instead of the Michaelis function, you can put higher order Hill activation here, or some more involved reaction. What we're going to discuss is not going to change whether this is a Michaelis equation or just a linear function, as long as it's a monotonically increasing function of x, so more xm, the larger this term, then all the rest will work just fine. Okay, so if we take our differential equation, which is rate of change of xm, the amount of methylated receptor, and that rate of change is the methylation effect of VRR, so the methylation effect of key R minus the demethylation effect of B. So VBB times XMK plus XM, which is our Michaelis term. So to be able to find the steady state XM, so we want steady state XM, we know that the rate of change of XM should be 0, meaning we can say this whole thing is 0, and then simplify. Obviously you can just multiply both terms with K plus XM, which would give you VRR times K plus VRR minus times XM steady state minus VBB XM steady state, which would then we can kind of move things around, meaning we can reparenthesize this as VRR times K plus XM steady state VRR minus VBB. So that's equal to 0. And then if we move this xm term to the other side, it becomes xm steady state VRR minus VBB. Actually, that's minus is equal to VRR dot times K. We could get rid of this minus by moving it inside. So instead of VRR minus VBB, we'll just do VBB minus VRR. And if I move that to the other side, XM steady state is essentially VRR times K divided by VBB minus VRR. So we can calculate the steady state like we want. Now that we have the steady state equation for XM, the amount of methylated receptor, we can find out the activity, rather derive the activity from that. Now we'll assume that a fraction of the receptors, the methylated receptors, are unbound. They have no ligand attached initially. Actually, this is generally the fraction of receptors that are unattached, because the attachment doesn't kind of is independent of methylated and unmethylated. But at this point, we're interested in the methylated part anyway, because those are the ones that are active. So, for example, if the amount of sugar is high, A0 is going to be low. If the amount of sugar is low, A0 is going to be high, because we know that the active receptors are the ones that are unbound. So you can come up with the tumbling activity at time 0, the initial steady state, as number of methylated receptors at the steady state times what fraction of them are active. That's the activity, the tumbling activity, because the active unbound receptors essentially determine the amount of key Y phosphorylated in the cell, which determines the tumbling activity. Now let's assume somebody adds attractant, sugar, amino acid, something to the environment of the bacterium, and suddenly the sugar concentration or the attractant concentration increases. Now suddenly the fraction of methylated unbound receptors becomes less, because that attractant is going to come and bind them. So now we have a new A1, which is smaller than A0, and therefore the activity, since A1 is smaller than the A0, the activity is also smaller, because XM initially is the same as the original XM steady state. It will adapt in time, but right now the activity suddenly drops to capital A1, which is much lower than A0, and the bacterium essentially stops tumbling as much. In time, as XM reaches its new steady state, hopefully you'll get the original tumbling activity back, if the adaptation is exact. And the way we can calculate this new steady state, XM steady state prime, so to speak, is by calculating a new rate of demethylation for key B. Because remember that the moment the activity of A drops, key A drops, the amount of key B that's phosphorylated will also drop, and this is almost instantaneous when compared against the methylation speeds. So initially, suddenly, when you add more attractant, key A becomes less active, which means key B will shift towards dephosphorylated, meaning VB will slow down. So we represent this with a new VB called V prime B, which is a slower demethylation of XM. We can so calculate the new steady state XM steady state prime, which is a steady state the system will arrive at once the methylation adapts in terms of this new V prime B. Now for exact adaptation, A0 and A2, capital A0 and A2, should be the same because we want the activity to remain the same once the bacterium has adapted. That means the amount of active receptors, unbound receptors, the fraction of unbound receptors times the initial steady state should be equal to the new fraction of unbound receptors, which is lower now that there is more attractant in the environment, times the new steady state should be equal. So if we plug in the equations we found before for the steady states, or before and after, and we assume, for example, that initially the activity of the receptors, the percent of unbound receptors was 100%, and later on when the sugar increased, only 10% are now unbound, and we assumed some trivial values for K, the initial VRR, and the initial BBB. We find that the activity initially comes out to be 1. Now we want the second activity, the final steady state activity, to be also 1, which means we have to solve for the new VBB, V prime BB, which turns out to be 1.1. Now the problem with the fine-tuned model, and the reason it's called the fine-tuned model, is that it stipulates that the VB and the change in VB is fine-tuned against what VRR is. Remember that the steady state level of methylated receptor is a function of VRR and VBB, and we essentially solved for VBB by setting VRR to, in this case, 1. The problem with that is it's not robust. It's not robust in two ways. Changes in the amount of B or the amount of R in the cell would actually cause, firstly, a change in the steady state rate of tumbling. So just the initial steady state would be different as well, but also more importantly, it would disrupt exact adaptation. So let's say our particular bacterium has maybe 80% of some average level of R inside the cell. So we know that there's about maybe 100 molecules of R on average in each bacterium cell, but this is not an exact number. It's a distribution. Think of it as a normal distribution, a curve. The average might be 100, but some bacteria might have more, some might have less, and let's assume you're a bacterium that has a bit less, maybe 80 molecules of it, so concentration drops by 20%. Now, the first thing that happens is when you change the VRR, so we're assuming VR stays the same, but the concentration of R is 80% of what we initially fine-tuned the model to. VRR is now not 1, but 0.8. So when you solve for it, you find out that the initial steady state is no longer 1. It's a bit less, so the bacteria initially even tumbles less. But what's more troubling is that when you increase the amount of sugar, namely when you decrease the number of unbound receptors, the percent of unbound receptors down from, let's say, 1 to 0.1, the new steady state is even lower, and more problematically, it's not the same as the initial steady state. So you've lost exact adaptation. That's the problem with the fine-tuned model. It's good on paper, but it doesn't behave the way actual bacteria behave, in that they are robust to this kind of change. We'll see later on experimentally that yes, the steady state tumbling rate changes when you change R, but real bacteria don't lose exact adaptation when you play with the amount of R. So this model does not really exactly explain what's happening. It doesn't explain the robustness of these bacteria to changes in R or B for that matter. The fine-tuned model was introduced around mid 80s, 1985 or so, by Goldbetter and Siegel. And at the time, like I said, it looked good on paper, and it did explain how this pathway worked to some extent, but obviously it wasn't explaining the robustness factor that well. So we know for a fact that bacteria are robust in their exact adaptation with respect to changes to the basic levels of the key proteins. So what happened was later on in the late 90s, a new model was introduced, the Barkai-Liebler model, which made kind of a theoretical assumption that the adaptation should be a function of something else, not necessarily the amount of methylation on the receptors, but it should be directly coupled to the activity. And they introduced the model by stipulating that, by the way, at this point it wasn't known experimentally that this was the case, or it wasn't shown structurally, but Barkai and Liebler essentially assumed that to be robust, the action of key B should only be on active methylated receptors. So if a receptor is actually bound by a ligand, for one thing, it inactivates. For the other, but the other part is it changes conformation so that key B can no longer access its methylation sites. So once a ligand is attached, it can only that receptor can only be further methylated. It cannot be demethylated until the ligand detaches. So we get the picture shown on the slide. The net effect of this is that now in our differential equation, the substrate term for B, so VBB times, remember originally it was the Michaelis term for XM by itself. Now the substrate term is a Michaelis term on only the active XMs, the unbound XMs. Remember that the lowercase a we used to represent the fraction of receptors that are unbound by ligand. So now it's slightly different, and also the activity is directly activity, by which I mean the activity of tumbling, is directly the same as the substrate we're using, which is A times XM. Now notice that when we take this form of the differential equation and we solve for the steady state, make the rate of change equal to 0 and rearrange, we arrive at VRR is equal to VBB times the Michaelis term for AXM. Now, it's interesting because now VRR is dependent on the steady state is dependent on what VRR and VBB are. So if it will always stay the same, the steady state itself, the value of the steady state activity will depend on what VRR and VBB are. So for example, if VRR is higher than normal, then the steady state will be higher than normal. If VBB is higher than normal, the steady state will be lower than normal. But it doesn't. Since we've added the activity of the receptor directly inside the function, it no longer depends on activity. So it will adapt exactly whatever the values of R and B are. So let's elaborate on the mechanism here for a couple of minutes. Now, our differential equation is always of the form rate of change of the methylated receptor, so that's our control system. We control our sensitivity based on how many of the receptors are methylated. Has a methylation term, which is VR times R, so the methylation activity of R, minus VB times B, the demethylation activity of B, and since B needs a substrate, there's the Michaelis term normally here, but for now, let's just generalize this and say some function of xm. In our case, this function of xm is simply xm k plus xm, or if it's Barkai-Liebler, it's axm over k plus xm. So if I go for the steady state, right, if I make this 0, then it becomes obvious that if I rearrange this equation, that VRR over VBB is always going to be equal to f of xm steady state. So keep that in mind for a minute, and then let's retake this original differential equation and take a look at it. So d of xm dt minus vrr minus vbb times f of xm. Now what I can do is essentially divide both terms with VBB. So if I do that, this becomes equal to VRR over VBB minus f of xm. And let's actually put this outside here so that we don't break the equation. So the rate of change, I haven't basically changed anything, just did a rearrangement. What's interesting though is this part. Notice that this is exactly the steady state. So now the rate of change of xm dt is going to be equal to VBB times f of xm steady state minus f of xm. So if you think of the rate of change of the response, so think of it as like your iris opening and closing in response to light, or the bacteria methylating or demethylating in response to changing levels of attractant, you're basically, it's proportional because of this VBB to the difference between the steady state and the current state. So what it's actually doing is taking the error and moving the control in that direction. So if the steady state is higher and the current state is lower, so the current state has a deficit with respect to the expected steady state, the error term will be positive, which means the rate of change will be high, will be positive as well, meaning it will push the system towards the steady state. If it's the opposite, if f of xm, the current state is higher than the expected steady state, then this term will be negative, the error will be negative, which means the rate of change will be negative, pushing the system to lower methylation to adapt. And notice that this will never stop until the rate of change is 0, and that will only happen when the error is 0. So to get, essentially, it forces constantly the system towards the steady state. Now, this is what's called integral control in control theory, or integral feedback, because what it's doing is it's constantly taking essentially the integral of the error, because it's a rate of change. So in time, the level of xm is the integral of this rate of change, which means it's adding up responses to errors either up or down, and it will not rest or not stop correcting until all the historical error has been zeroed out. So as long as there's kind of the total error over time is not equal to 0, this system will not stop adapting. And that's exactly what we want. We want it to stop when the error is 0, which means it has adapted completely. Now, those of you from an engineering background will notice this is, and if you've taken some kind of process control or control systems, whatever, you'll know that this is essentially a PI controller, a proportional and integral controller. And we know that when there's only proportionality in the control, so the response is just proportional to the error, but it doesn't add up the error, you still get decent control, but you get a steady state offset, meaning your steady state, when control stops, the point at where you're left is not exactly the set point, which is handled by the integral term. So when you have integral feedback or a PI controller, you know that your system will always come back to the steady state, or in engineering parlance, the set point. So now, what's different between our system with in Barkai-Liebler versus fine-tuned? We'll look at that, but before that, I have something to say about the form of this function xm. Now xm, let me delete this part. We don't need that anymore. Xm can be any function that's what we call monotonically increasing with x, for in this case xm. So any function essentially that goes up as xm goes up strictly, and it goes down as xm goes down, is a function that's required here. So for example, the fine-tuned models, Michaelis function, when you look at it, it's something like this. So if we're using f of the fine-tuned model, which is this thing here, with xm, it's going roughly like that. We could just use a very simpler model, like f of xm is just equal to xm. And that would be a first order reaction now, not a Michaelis reaction. That's also fine, right? So simple versus xm would just be like that. It's fine. You could use a higher order Hill function. That's also fine. It would go something like this. And that's also monotonically increasing, because you know that there's no point in which, if one value of xm is higher than another value of xm, the function will never be lower than its previous value. So it's increasing. What you can't have is, well, something like this. For example, goes up, and that is not allowed. And you can see the reason, because if that's allowed, this error term here will cause that function to kind of get stuck in this value here. Because at one point, it will, if you push xm, it will come in here, and if it goes up, error goes up. If it goes the other way, error goes up. So you'll get a steady state xm here, which might not necessarily be the right place. So what makes Barkai-Liebler different than the fine-tuned model in this respect? When you look at it, the fine-tuned models, let's delete this. The fine-tuned models function is 1 over, xm over k plus xm. The Barkai-Liebler function is more or less the same thing, but that's activity times xm, k plus, well, not activity, sorry, this is the fraction of unbound or active methylated receptors, a times xm. So these two are basically the same function. It's just that, remember that in this case, if I do a little renaming, I can rewrite the Barkai-Liebler model as xm star, let's say, which is, let that be a stand-in for a times xm and xm star. So this would be a function of xm star. Now, what this one's doing is, now the methylation is looking at the error between xm star steady state minus the current xm star, and this is the quantity that we're trying to zero the error out against. Whereas in the fine-tuned model, what you're trying to zero the error on is purely the methylated things. So notice that this xm star is exactly the tumbling activity. So we're trying to zero out, once the control is done, we're trying to zero out the error on activity, not just methylation, as in the fine-tuned model. As an analogy, think of so actually the control system and the integral feedback is there. It's just looking at the wrong thing. As an analogy, you can think of something like this. Assume you have a cliff and you have two levels on the cliff, and there's a crane here. And on the crane is a motor that has a rope that goes down and has to bring up a weight from, let's say, it's not a cliff, but a building from ground 0 to ground 1, and you know the distance between ground 0 and ground 1. And what you can do is kind of add a control system to this motor that will monitor power, wattage, right? So it will see, essentially be the rate of change of, let's say, the power I'm giving this motor should be some constant times the power needed to reach floor 1 minus the current power, or something like that. So what this will do is, if you calculate this and fine-tune this P1 correctly, when you run this motor, it will just spend just enough power to get this weight up to here, which is fine, but it's not exactly what you're trying to accomplish, right? You're not trying to save power or optimize on power or whatever. What you're trying to do is make sure that this thing gets from 0 to 1, so it moves this distance of h in the upward direction. And you can see the problems with this if you think of the same problem, but now somebody comes in and attaches a larger weight. Let's say a weight that's 20% larger to this motor. What's going to happen is your system will try to get to this power, to the original fine-tuned steady state, fine-tuned set point, but since the weight is more, it requires more power to bring this weight up the same amount of distance, obviously. So what will happen is it will stop midway. So that's the analog of the fine-tune model. It assumes, and the metaphor, the analog here is that the size of this weight is VRR. It assumes that it will stay constant, but if it changes, then your set point is no longer valid. So in this motor example, what you would do is you would actually build the error term on essentially the true value that you want to control, which would be height of the first floor minus the current height. In which case, if the current height, as long as the current height is smaller than the height that you've reached so far, you would actually keep adding more power. If you overshoot somehow and go past that first floor, now this term will be negative, which means you'd have to run the motor in reverse for a certain amount of time to get it back to exactly the right floor, which means this is the right quantity to be looking at the error of, rather than the amount of power or the work you've done, because that's sensitive to what the size of the weight is. Same thing for chemotaxis models that we've looked at. Fine-tuned looks at how much it has methylated against a set point methylation, whereas the Barkai-Liebler model effectively looks at the true quantity of what you want to control, which is tumbling activity. It makes sure that the tumbling activity returns back to what it's supposed to, regardless of what the other factors are, in this case, the amount of VRR. It's not a surprise then that when we simulate with the Barkai-Liebler model, and hopefully I'll record a separate session showing you how to implement this, we get exact adaptation, whether VRR is 1, like as in the fine-tuned example, but also when we have VRR is equal to, for example, 0.8 in this figure. Notice that there's a difference between the two responses. When VRR is less, the actual steady state is lower. So you have a bacterium that's less likely to tumble in the normal state of affairs. The bacteria that has more R is more likely to tumble when it's adapted, and the response sizes are slightly different. But other than that, the exact adaptation is the same. At the end of the plot, it hasn't quite adapted yet, but just take my word for it. If you follow the plot past 1,000, it will converge to the initial steady state. It's interesting that we actually see this difference in different bacteria. When you look at these bacteria under a microscope, you realize that some of them are more nervous, as they call them, than others. So within the same community, one bacterium might be more likely to tumble. Another one will be less likely to tumble, so a more calm bacterium versus a more nervous bacterium. And it's assumed that these differences are due to the differences in their internal expressions in R, and it actually confers an evolutionary advantage to the colony, in that or the group, in that the nervous ones will react faster. They'll have more sharper reactions, but then the other ones will be less reactive, and even when there's sharp gradients of sugar versus shallower gradients, some part of the group will be well adapted to it. But regardless of whether they're nervous or calm, all bacteria always come back to their original steady state, so that they all have exact adaptation. Barkai and Liebler's group have actually done experiments on this in the following years after their model, and the first experiment actually checks whether bacteria really do adapt back to the exact steady state they were. So in this figure, you can see at the top the points that are little squares in the top curve. Those are observations for 20 minutes of just normal wild-type bacteria, E. coli, and their tumbling was tracked with video tracking. I think it's a group of 100 to 400 bacteria, and as you can see, they tumble at a relatively steady state of about 4.4 to 4.5 tumbles per second. So they tumble almost once every 2 seconds. And these were bacteria that had no attractant, so they were unstimulated. They also tried this with stimulating the same type of bacteria with attractant aspartate at time 0, and then measured their tumbling frequency in time. So that's the second curve that starts from the bottom with little circles as the data points. And as you can see, at time 0, when the attractant was added, there's almost no tumbling. They tumble less than once every 20 seconds, and they're just swimming directly. But in time, about halfway point is about 10 minutes. They have halfway adapted, and by 20 minutes, they're back to where their unstimulated cousins were, meaning they adapt to the exact same steady state tumbling frequency on average. In order to test the robustness of actual bacteria with respect to changing levels of key R, the R enzyme, remember that the one that methylates the receptors, they actually knocked out the natural key R gene from a strain of E. coli, meaning they erased that gene so that the bacteria could not produce that gene anymore by itself, and they kind of engineered the plasmid that had a copy of key R, which is a separate kind of piece of genome, and which was promoted by a light-sensitive promoter. So, and then they it kind of infected these bacteria, the knocked-out bacteria, with their own version of the plasmid of key R, and that way they could control how much bacterium could express the R protein by varying the amount of light that the bacteria was growing in. So more light means the bacteria produces more R on average, and less light means it produces less. So in this graph here, you can see in the x-axis the fold change of R. So 1 is about the amount of R produced by the wild strain, normal bacteria, and they've tried from about half the normal amount up to 50 times the normal amount. So that's a fold change of 50. And as you can see, for a very wide range of R, for R expression, bacteria still were able to exactly adapt back to their original steady state, within error bounds, of course, but this is pretty impressive how the system can actually perform exact adaptation even in kind of dramatically different amounts of a critical protein, in this case R. Finally, they looked at the change in adaptation time and the steady state tumbling frequency of these bacteria as the amount of key R expressed by these bacteria changed. So the black dot is roughly the wild type bacterium. So on average, it's tumbling at 0.4 or 0.45 tumbles per second, and the adaptation time for a 1 millimolar step increase in aspartate concentration is about 10 minutes. Now notice what happens when you reduce R. The little square data points are the tumbling frequencies for different fold expressions. And the right side scale is the values for that. So when you're going down to, for example, half the amount of wild type R expression, adaptation time, the tumbling frequency drops down to about 0.2 per second. And notice that the adaptation time for the same bacterium, which is the circle at the same fold expression, is above 20, close to 25 minutes of adaptation. So when you reduce key R, the bacteria take longer to adapt, and their overall tumbling frequency, their steady state tumbling frequency, is less. On the opposite end of the scale, if you look at something like 6 times the normal amount of R, the tumbling frequency is increased to, the steady state tumbling frequency is increased to 0.6 tumbles per second, and now they adapt within a couple of minutes. So you can see again that as you have more key R expression, higher amounts of key R in the bacterium, it becomes more nervous, and as there's less key R, it becomes a calmer bacterium. And in nature, obviously, there's a distribution of these in any group of bacteria, with the kind of the wild type point here of steady state tumbling frequency of 0.45 or so, and an adaptation time of 10 minutes being the center of that curve, of that distribution, the mean of that distribution. Okay, so this is all about robustness in bacterial chemotaxis. Hopefully by next week I'll put out another video explaining kinetic proofreading as another example of robustness in biological systems. And in the meantime, I hope to also put up a video of how you would implement both the fine-tuned model and the Barkai-Liebler model using Python and SciPy, and plotting the results, etc.